Okay. All right. So I'm Kuldeep Bansal. I'm currently working as a researcher at Akademi University. And before moving to Finland, I did my PhD from University of Nottingham in England. And I'm really glad that my supervisor is also going to talk. My PhD supervisor is also going to talk tomorrow. His name is Professor Cameron Alexander. So the work I'm going to present today is have some hint of my PhD work. So during our PhD, we are making polymers for drug delivery, and then we realized that if we are able to make a polymer which can be functionalized based on the desired application, it will be great. So when I joined a postdoc here, I was working on that, and fortunately, we figure out how to do that. So today I will talk about this polysjasmine lactone based polymers and what they are, how they have synthesized, and in what kind of applications we are able to use them. So the idea start with the polymer drug conjugate. So basically, the only polymer in the market coming as a polymer drug conjugate is polyethylene glycol. But the problem with polyethylene glycol is like it's having only one functional group to attach the desired drug or other therapeutic moieties. So basically, the drug loading remain low, and that's why there was research going on where we can increase the drug loading by increasing the number of functionality of functional molecules on a single polymer chains. Some research are ongoing with the polymer dextran, poly-HPMA, or polyglutamic acid, but still none of the polymer make its route towards the market. So there is still a scope to develop polymers which can be synthesized easily and functionalized easily, but can also be used for different applications. So we come up with the solution of making the polymer using jasmine lactone, a monomer which is already approved by FDA as a flavoring agent. And by using simple chemistry of ring opening polymerization, we can able to make the polymers with the different properties. For example, we make homopolymer, which is hydrophobic and amorphous in nature. You can see here the homopolymer, which is quite amorphous. Or we can use initiator as a polyethylene glycol, which provides the hydrophilicity to the polymer. So we get a block copolymer, amphiphilic block copolymer, which can also be self-assembled into the micelles. The beauty of this polymer is it's provide this double bonds at the side chains, which can be functionalized to different functionality. So we utilize this opportunity and converted this polymers into different functionality. For example, we insert carboxylic functional group, hydroxyl functional group, or amine functional group by a simple thiolene click chemistry in the presence of UV light. The reaction is straightforward. The purification is very easy. So it's make a way to make polymers via easy synthetic methodology. So as we decided to use this polymer first in polymer drug conjugate, so we move forward and selected one derivative of the polymer, that is hydroxyl terminated polymer, and attach doxorubicin as a model drug on it. So first we converted the doxorubicin into the doxorubicin disulfide molecule. So why we choose disulfide here is that doxorubicin as an anti-tumor agent, but it is very toxic to the heart cell. So it has been reported in several publications that tumor cell having the high amount of glutathione, which is responsible to cleave the disulfide bond. So it was assumed that when this polymer drug conjugate enter to the tumor cells, that this disulfide bond has been cleaved and the dox will release, which give the therapeutic response. On the other hand, when this polymer conjugate enter to the normal cells, because of the low level of glutathione, this cleavage is not as much as we can found in the tumor cells, so which can reduce the side effects. So we then move to do some physical characterization where we have checked the size by DLS or by TM. So the surprising things we observed here is like even after reconstitution of this polymer drug conjugate, we haven't got much aggregates or or any difficulty to reconstitute. We haven't used any cryoprotectant here, so that's make it very promising. So before freeze dry, the size was around 40 nanometer, and after freeze dry, the size was around 55 nanometers. So only a couple of nanometer has been increased. There was also a second peak, which might be belongs to the aggregates, and then PDI is also not very high. Then we check the drug release in the presence of dithioerythritol, which is the source of uh, reducing environment. So in the presence of 10 micromolar of DTT, we have seen a very slow release and only around 15% of the drug release after 9 days, while when we increase the concentration up to 1 millimolar, the release has been accelerated and it goes up to 40% after 9 days. Then we treated this polymer drug conjugates to the tumor cells. We choose here MDMB 231 cells. And at different concentration, we try to find out whether it's really producing the glutathione responsive release and the activity or not. So on the extreme right hand side, these are the free polymer with different concentrations. So up to 2 milligram per ml, we haven't seen any significant toxicity compared to the control groups. This is the free doxorubicin, so which indeed is quite toxic, and up at 50 microgram per ml concentration, it kills almost like 20 kills almost 75% of the cells. While on comparing with the polymer drug conjugates in the presence and without the presence of glutathione, you can see a clear difference in the cytotoxicity. So when we have a glutathione, that will be responsible to cleave the disulfide linkage, which activate the drug, and thus we have seen a higher toxicity compared to the absence of glutathiones. We move further to check this on FEX confocal to see whether they are entering inside the cell and if the drug is activating or not. So we first run both of the free doxorubicin and the polymer conjugated doxorubicin, and you can clearly see here that when the drug is conjugated with the polymer, the fluorescent intensity has been significantly reduced. So that is the key we have decided to evaluate. So when there is a presence of glutathione, the drug will release or cleave from the polymer conjugate, and then its red fluorescence has been increased. So you can see here in the presence of glutathione, this is the free doxorubicin, quite fluorescent, while in the presence of glutathione, you can see some fluorescent, but in the absence of glutathione, the fluorescent intensity is less compared to the fluorescent intensity when we monitor it in the presence of glutathione. So that's clearly suggested that the drug is releasing in the presence of glutathione to a greater extent as compared to the absence of glutathione. So this is how it makes stimuli sensitive drug release. Further, we checked the same thing quantitatively with the FEX, and again in the presence of glutathione, there is higher fluorescence as compared to in the absence of glutathione. So this makes sense that when we have a glutathione, the disulfide bond cleaved, which activate the drug, and we can probably see a higher toxicity as compared to the absence of glutathione, which is the case in the normal cells. So this research has been published in 2021, and after that, we also move and take this polymer conjugate forward to the in vivo study. So we treated to the 41 tumor models. The dose we have used was 7.5 mg per kg. And we have 4 different groups. So one is control, one is free doxorubicin, the another one is the liposomal formulation of doxorubicin called altrizome, and then we have our polymer conjugate. So we start with the by seeing the weight change in the animals, and you clearly see in the presence of doxorubicin or altrizome, there was a sharp decline in the weight, which clearly indicate the toxicity, while the control and the PGL dox doesn't shows any toxicity in terms of weight change. Then we monitor the tumor volume here up to 14 days. So at the day 6, we start injecting the samples, and after up to 14 days, all the animals has been killed in the group of doxorubicin or retrozoam. So clearly again indicating the toxicity. You can also see here in this picture. So these are the mice treated with doxorubicin. Hair loss, weakness can be easily identified, while the polymer conjugated mice are more healthy. In terms of reducing the tumor volume, we have seen a significant difference compared to control group, and the survivability will also increase up to 40 days. However, as expected, we were believing that the tumor regression should be much higher as compared to the control group. But if you remember, in the release study, we have observed that even after 9 days, we got only 40% of drug release. So incomplete drug release might be the reason that we haven't seen a great regression in the tumor volume compared to our polymer drug conjugate, and that can be uh, described by the structure of this polymer conjugate. So it makes micelles where the drug has been conjugated to the hydrophobic region, and glutathione being the hydrophilic moiety, it might have the difficulty penetrating deep inside in the core of the micelles structure to make the drug cleaved from the disulfide linkage. And that's why most of the research mainly making the disulfide linkage at the edge of the hydrophilic and hydrophobic polymers, so that they will see a quick release. But nevertheless, we change our approach now, and we have now decided to use thioketal linker instead of disulfide linker. So here we use the acid terminated polymer, and then choosing the diclofenac as a model drug, we conjugate it with the thioketal linker, which is reactive oxygen species triggered linker. So in the presence of oxidative environment, this bond has been cleaved, which allowed the release of drug. We did quick study on the release, and in the absence of oxidative source, here we use KO2 as a oxidative source. In the absence of it, we hardly see 2% drug release after 2 days, while in the presence of KO2 1 millimolar, the drug release has been accelerated, and we got almost 85% of drug release after 2 days. So we basically able to overcome the incomplete release issue by changing the linkers, and because this ROS sensitive formulation can also be used in Alzheimer or arthritis, because they are also responsible for generating a high amount of reactive oxygen species. So these are not only limited to the cancer disease. But next, we prove that our polymer can be used for different linker chemistry. We are also very interested to see if they will able to make micelle and encapsulate the hydrophobic drug like this Genexol PM developed by Samyang Biotech. So we start with the acid terminated polymer. So we use amphiphilic polymer having COH group, and then we compare it with the different polymer, for example, marketed Sol Plus or polylactic acid amphiphilic polymer, or unmodified polygjasmine lactone, or having the polygjasmine lactone with a phenyl or aromatic group at the end. What we have observed in this study, that in the presence of acid functional polymer, the basic drug dissolve at much higher rate. For example, in cinnarizine or clytromazole, you can clearly see a difference in the amount of drug solubilized in this micelle as compared to the other formulations. But indeed, in one case, in the case of estradiol, Sol Plus was found to be better as compared to any of the formulations. So there is still a chance that we can probably use the hydroxyl functional polymer and see how it will encapsulate estradiol. So it makes clear sense when there is a acidic polymer and the basic drug, it will make an ionic interaction, and we get the higher encapsulation. We continue this study by using some more drugs, where we use weakly acidic and the basic drugs, and the results are same. The weakly acid drug did not encapsulate well, while the weakly basic drug with the acid terminated polymer show higher encapsulation efficiency in this green bar. So now there is a strong ionic interaction between the acidic polymer and basic drugs. We have to prove that this ionic interaction is not hindering the drug release. So we did quick release studies at different temperature and pH, and what we have found that at room temperature at basic pH, after 7 days, we saw observed the sustained release where only around 25% of drug has been released after 7 days. But at the acidic pH, the drug release has been accelerated because this COH functional group get ionized, and then which weaken the ionic interactions. So that makes sense that at acidic environment we can able to get the higher percentage drug release. So around 75 78% of drug release has been observed at the acidic pH. Then because we haven't checked the cytotoxicity of acid terminated polymer in the previous study, we checked the OH terminated polymer. So here we did a quick cytotoxicity study and we compare our results with the pluronic P123 micelles. So up to 250 microgram per ml, there was no toxicity has been observed, but when we increase the concentration up to 500 microgram per ml, there is a sharp decrement in the cell viability with the pluronic sample. So here we can clearly say that the polysesmin lactone based polymer are safer as compared to the pluronic P123 micelles. But as we increase the concentration up to 1 milligram per ml, the COH terminated polymer also starts showing the toxicity, while the pluronic P23 kill almost all the cells. So yes, so now we are currently in the progress of finding the way to commercialize this polymer. So we already running a project called Jasmine Pro, which is funded by Business Finland. So these are our team, and you can also like visit our website to get a little more information. Website is still in development, so you probably won't get much information, but feel free to contact if you need some information. Thank you, and I will be happy to take questions.